Hello everyone, I am Dibyendu Samanta from IIT Kharagpur, and today I am going to discuss about the last lecture of this module, module 1. Here I am going to discuss about mechanistic overview of DNA replication. During my last lecture, mostly I concentrated on the basics of DNA replication, how Mechanistic and Stahl they proved that DNA replication is semi-conservative, but here mostly I will focus on the mechanism of DNA replication, how it actually works. So, I must say here that the mechanism of DNA replication is very complex. There are many problems, there are many challenges, and accordingly because of long time of evolution, different life form, I would say in living system, they also evolve with this problem so that they can take care of this, because DNA replication is a very fundamental step in life. So, now in the first place I am going to discuss about the template and raw materials for DNA replication, and then the more complex part, the mechanism of DNA replication and associated complexities. So, there are many enzymes, they are also involved, many different complex nomenclature, but I will try to keep it very basic, fundamental idea, and then if you are getting more interested about this idea and this field, then you can refer any standard textbook. So, here Arthur Kornberg, so he received Nobel Prize in 1959 because of his phenomenal work on DNA replication. So, particularly he discovered the mechanism of biological synthesis of DNA, how DNA is getting synthesized by DNA polymerase. It is an enzyme, DNA polymerase. This enzyme name, they are very much logical. Most of those names are very much logical in biological world. So, most of the enzymes they end with ASE, as polymerase. So, polymerase means some enzyme which will make the polymer. What kind of polymer? In this case, DNA polymerase. So, they make the DNA polymerase. So, as a result of that, the function of DNA polymerase is they will add deoxyribonucleotides one after another, and they will make the DNA polymer. So, now again Arthur Kornberg worked with E. coli, because E. coli is a model organism. It is a simple bacteria and it has just one DNA molecule, one circular DNA molecule. This is the difference between our DNA and bacterial DNA. So, bacterial DNA, they are circular, but in case of human or any eukaryotic DNA, they are linear DNA, and also in case of E. coli, they have only one DNA molecule or one chromosome, whatever we say. Now, what are the ingredients required in order to carry out DNA replication? So, to understand that, so I would very briefly, if I say there are 4 major components that are required for DNA replication. So, what are those 4 components? So, if we understand properly the DNA structure, what are present inside DNA, then we will be able to provide answer to this question. So, I would say the first component should be the raw material, which is which constitute DNA. So, those are the deoxyribonucleotide triphosphate. So, I am just writing here DNTP. So, deoxyribonucleotide triphosphate. So, that means here it can be DATP, A stands for adenosine here, DGTP, DCTP and DTTP, because A, T, G, C, those are present in DNA. So, those are the 4 major ingredients, raw material that should be required for the DNA replication, DNTPs. Then, as we know from Mechanistic and Stahl's experiment that it is semi-conservative, that means we need a template, and from that template we will get the new strand. So, as a result of that, here also we need, for the replication, we need DNA template. Those are the basic requirement for DNA replication. DNA template is required. And so that we can copy the information what is present in template, we have to make the newer copy. Now, some enzyme is required which will actually synthesize this DNA or synthesize this polymer. The name of this enzyme is DNA polymerase. So, first DNA polymerase was isolated, I would say purified and characterized by Arthur Kornberg, and that is why the name of that enzyme at that time, it was commonly called as Kornberg enzyme. And then the last but very important thing is magnesium ion. So, magnesium ion is also very crucial for this DNA replication step, because DNA polymerase cannot function without magnesium. It requires magnesium for its function. So, these four are absolutely required for DNA replication. As I already told, this is just for fun that this car actually belonged to Arthur Kornberg. So, he was so obsessed with protein purification, particularly purification and characterization of DNA polymerase, so that he can understand the replication mechanism. So, if you can see, the number plate of his car here is purify. So, he is so obsessed with the purification of enzyme, particularly DNA polymerase. So, this is in California. So, California, and then followed by the number here, special number purify. But anyway, that is not that important. Now, the chemistry of this reaction, or this how it how DNA polymerase actually polymerize. So, the polymerization of nucleotide triphosphate by DNA polymerase require free 3' OH group. This is very important, and that is why I told several times that any nucleic acid, whether it is DNA or RNA, it always extended from 5' to 3' direction. Again, I will discuss here. So, as you can see, so this is an existing DNA strand. So, here in this existing DNA strand, you can see that 5' like this is 5' position, this is the 5' and why? Because in this strand, the 5' carbon of this sugar is capped with the phosphate group. And here if you see in this, here this is the 3', because the 3' carbon of this sugar is with the OH group here. So, this is free to react. So, only this DNA can grow in this direction. That is why always I say that 5' to 3' extension. But why in this extension? Soon you will know. So, now what will happen? This OH group available at the 3' end of this existing DNA strand, this OH can act as nucleophile, and it will create some nucleophilic attack on this incoming nucleotide. In this case here, as you can see, this is GTP, right. So, here I already mentioned that you can see the guanine base here, and then this is the sugar, and this is the triphosphate. So, this is GTP. So, now because of this nucleophilic attack, you can see that 2 phosphate here, the diphosphate, it is getting released, and now you will see a new phosphodiester bond here. So, as a result of that, now in this existing DNA, we have 3 nucleotides A, C and T, but here this is longer DNA. Whatever we had before, now we have one extra nucleotide here. Here this is G residue or the G nucleotide added here. But if you see at the end of this G residue, again you have the 3' free OH group. So, again one nucleotide can be attached here, and that is why DNA or any nucleic acid, it does not matter whether it is DNA or RNA, the basic mechanism is same. It is always grow from 5' to 3' direction. But now the question is, why this G is coming here? Why not A? Why not T and C? So here for the explanation, I am just showing here that G is getting added here, and you have G residue already added. But this is template dependent synthesis. So as a result of that, I would say that here this is the 5 prime, this is 3 prime. So you have here T, G and A. And if this is so, then in the growing chain here, as you can see, this 5 prime, A, C and T. The same sequence I am writing here, that A, C and T. And then the next one here is G. That means the guanosine added here, the guanine added here. So as a result of that, since this is template dependent, that means in the template you must have here C, and that's why this G is added here. Similarly, the next nucleotide will come depending on what is present in your template. If you have G here, then the next one will be C, because inside the cell you have all these 4 nucleotides available in the surrounding, right? But it is the template sequence which is present here. So the template, because depending on the template sequence, the enzyme DNA polymerase will put the correct nucleotide. This is very precise thing. So DNA polymerase, it has very precise role here. So it will put the correct nucleotide always. But if you study in detail, you will find that sometime, like this is very rare, but sometimes some wrong nucleotide can be added there also. But fortunately, DNA polymerase, they also have the proof reading activity. So they can understand also that the wrong nucleotide has been added. So they will rectify that mistake. So there are many other mechanisms to correct the the information, so that if something is wrong during DNA replication, that should be corrected, because DNA contains the instruction of life. So every information actually present in DNA, and finally from DNA we will get the RNA and then protein. So as a result of that, the main information should be intact and should not be spoiled with some wrong basis or some kind of mutation, I would say. So this is the chemistry of DNA replication, how the next nucleotide is attached to the existing DNA strand and which is catalyzed by DNA polymerase, a specific enzyme which require magnesium ion. Now, we are going to discuss about the the first problem here, because whenever I am telling you that this is template dependent, I am just showing that here you have one template, that this is 3 prime to 5 prime. You have sequence here A, T, G, C, C, G, and the new strand as a result of that it will be G, sorry, this should not be G, this is my mistake. So this should be T, A, C, G. It's going like this, right? So this is 5 prime to 3 prime. But this is possible, like the way I am explaining very simple manner, that you have the single strand available as template. But inside our body, inside E. coli cell, everywhere DNA is double stranded. And we discussed that if you increase the temperature of DNA, it's kind of really high temperature, I would say 70 degree, 80 degree centigrade, then those two strands will be separated. But our body temperature is much lower than that. So as a result of that, DNA is always double stranded and double helical in our body. So then the problem is how we will get the single strand template in order to replicate, right? So as a result of that, I would say, if we draw here like this is a DNA molecule. So I would say this is 5 prime, this is 3 prime, 3 prime, 5 prime. DNA, those two strands are antiparallel. That's why we are writing in this way. Now, the thing is, like temperature is ambient temperature in our body. So as a result of that, hydrogen bond between these bases will not be broken, and you will not get the single stranded DNA in order to replicate. So as a result of that, we have some more complicated mechanism, and that will solve this problem. So as a result of that, what happened? So we should get this kind of form. So this is 5 prime, this is 3 prime, and this is 5 prime and 3 prime. So if you are getting this kind of single stranded form, then we can synthesize the DNA. Then how we can do that? If we pull apart these two strands of DNA, then like in DNA we have lot of turn. So as a result of that, it is not possible to like pull apart these two strands in this way. Then the front part here, the DNA will be tangled together. So it's a real problem. The DNA will be tangled there. And also, how we will pull that? So for example, if we believe that we have some enzyme, in the next slide I am going to explain that we have some enzyme, it's called helicase. So that enzyme will break this hydrogen bond between the bases of double-stranded DNA in ambient temperature. So the name of this enzyme is helicase. So if helicase work here and it actually destabilize this hydrogen bond and those two strands will be separated, that is okay, but still there will be some problem, because it is not just double-stranded DNA, it is also helical. Just they have kind of coiled to each other because of their helical nature. So as a result of that, we need to solve this problem also. So that's why we have another enzyme that is called very interesting name, that is called topoisomerase. It seems like very complicated name, but it's not actually very complex. See, the function of the topoisomerase is, isomer, right? So topoisomerase. So I'm telling that when we are trying to separate these two strands by this helicase, then front part of the DNA it will be tangled. So since we are drawing in this linear form of DNA, that's why I'm telling that. But if you now imagine that the bacterial DNA, E. coli DNA is circular, it will be more problem. We cannot separate in that way. So as a result of that, topoisomerase is one enzyme. So this is called replication fork, where you can see these are two strands which are getting separated. So this is called replication fork. That means here the replication will be happening, replication fork. So just ahead of replication fork, this enzyme will work. So enzyme will cut DNA strand and then they release the turn. So as a result of that, if that is happening just ahead of this fork, then helicase can work. So this is the function of topoisomerase. So the name topoisomerase, because the DNA double-stranded double helical structure, if their sequence is same, but one is like after function of this topoisomerase, they are getting relaxed. They are this turn coiling that is getting reduced. But if you see their sequence, their molecular weight, they are exactly same. So they are isomer. But what kind of isomer? Those two are like differing because of the topoisomerase action. Their topological problem has been solved. So they are getting like they are differing by their topology. So that's why this enzyme is named as topoisomerase. So topological problem is getting solved by this enzyme just ahead of the replication fork. So this is called topoisomerase, and together the helicase and topoisomerase, they work together in order to get single-stranded template at the replication fork so that DNA polymerase can synthesize DNA smoothly. So I'm going to the next slide. Here you can see whatever I explained, that supercoiled DNA is getting relaxed by topoisomerase and unwound by helicase, and some associated proteins are there also. Is not, I'm just explaining like very basic thing here, but many other small things are also involved here. So now if you see, this is the direction of movement of replication fork. So that right now the replication fork is here. After some time when replication will be happening, so I would say now we are seeing here, right. Now after some time I would say the replication fork moved here, right. So here already maybe DNA has been synthesized in this place, it is going like in this direction. So over the time that way from one DNA molecule you will get two DNA molecules, right. So now here this is helicase which requires ATP and some other proteins are also involved, and that actually break hydrogen bonds in between these two strands, and topoisomerase is also functioning just ahead of it to relax DNA molecule, and that way it solved the problem, so that in normal body temperature still we have single stranded template in order to carry out DNA replication. And some other protein which is called single stranded binding protein, those are also attached to the single strand, so that in this temperature again those two complementary DNA strand will not be coming together, just to help the process of replication. Now like I will discuss few problem and I will discuss their solution also. First one I told about the topological problem, and now the second problem is DNA polymerase needs a free 3' OH of a pre-existing nucleotide to extend the chain. As I already explained during the discussion of the mechanism how the next nucleotide is getting attached, during that time I mentioned that it requires a free 3' OH group, as you can see. So as a result of that we are trying to make the DNA, but from where we will get the initial DNA, the pre-existing DNA, so that the reaction can be carried out, because DNA polymerase cannot add independently. So as a result of that we need to solve this problem also. In the next slide I am going to discuss that. So here as you can see DNA should be like always I mention any nucleic acid should grow from 5' to 3' end. So as a result of that I would expect that this is the 3' end. So as a result here we can see that 5' to 3' it can grow like this, 5' to 3'. But the thing is I just mentioned that DNA polymerase requires pre-existing 3' OH group. So that's why DNA polymerase they don't start the replication. Instead of that we have some other specific enzyme which is called primase, as you can see the name here, primase. So primase. So that means primase is some enzyme, people actually named it primase, so that they can start the process, they can make the primer, but this primer is made up of ribonucleotides. So you were actually initially we were like discussing about the synthesis of DNA. Now we saw that at the beginning we are making the RNA primer, and that's why we are telling it primer, because this is not the final thing, this is primer. So for example when we paint some house or apartment, before putting the actual color we put something which is called primer, so that after that we put the actual color there. Similarly this is primer. So it is just starting the process, but finally we have to remove this thing, because this is made up of ribonucleotides and this is called RNA primer. So finally we will remove this by some specific mechanism, and it will be replaced by DNA itself. But in order to start this process we first need to put the RNA primer here. So this is 5' end and this is the 3' end of RNA primer, which is polymerized by the enzyme which is called primase. So as a result of that, now in this short segment of RNA primer you have free OH group at the 3' end. So as a result of that, now DNA polymerase can extend this chain. So as you can see here, now the DNA polymerase, in this case specifically this is called DNA polymerase 3. So you will see DNA polymerase 1, 2, 3, many things. Most of those things are named based on their sequential discovery, but it's okay if we just mention DNA polymerase. So DNA polymerase will extend this from 5' to 3' direction, and it is synthesizing this strand, and this strand is going like steadily in this direction, and this strand is called leading strand, as you can see here. Why leading strand? Because it is going very steadily, and then if you see here in this portion, the fork will move towards right this side after some time, then this strand will be extended like this. It's kind of continuously getting synthesized. So as a result of that, it will go in this direction. But now the other strand, it has the problem, because like always DNA is getting synthesized from 5 prime to 3 prime. So as a result of that, there is no option to getting synthesized from this to this, because 5 prime position present here and 3 prime should be here then, because DNA is antiparallel. So then it has to go 3 prime to 5 prime, but that is not possible. So as a result of that, there is some problem in this place. So as a result of that, what happened in this strand, the DNA strand synthesis happened in a different orientation. So here in this strand, you will see that one short primer here, this is the primer, is synthesized by primase, and then the polymerase will be extending this primer towards this direction. So this is a short fragment. Why short? Because this end is not yet available. If this end is available, then one primer can sit here and it can go in this direction steadily, just like leading strand. But this end is not yet available. So it's still double-stranded and helical structure. So as a result of that, this strand will be synthesized in a sequential manner. So as a result of that, you can see another primer came here and this short segment of DNA will be synthesized. Then another primer will be there. So then when the fork will move in this direction, then another primer will be attached. So it needs multiple small primer here, and then small segments of DNA will be synthesized. And those small segment of DNA is called Okazaki fragments, based on scientist name who discovered this, Japanese scientist Okazaki. So he actually discovered this mechanism here in this lagging strand synthesis, and that's why those are called Okazaki fragments. So this way you can see the leading strand and the lagging strand. Lagging strand, because this is called lagging strand because this is lagged behind, because it is getting synthesized little slower than the leading strand, and it requires multiple primer, and in a sequential manner it is getting synthesized. So this way both the strands will be synthesized. But now the third problem is RNA primers need to be removed, because our goal was to synthesize DNA. This is DNA replication. So the thing is, there is again another specialized enzyme. It's called polymerase 1, DNA polymerase 1 actually. So it has inherent RNase H-like structure, like activity, I would say. So that is, this enzyme, whenever it will see some kind of DNA RNA hybrid, so you can see this is RNA. So now this is DNA. So DNA RNA, they have base pairing here also. So whenever it will see DNA RNA hybrid, it will destroy the RNA part. So as a result of that, the RNA segments, the RNA fragments will be removed from this lagging strand. So as a result of that, also from the leading strand. So as a result of that, no more RNA here. But now if you see, the RNA is not there, but DNA is present here. So this is one Okazaki fragment here. So as a result of that, here you have the 3' OH available. So as a result of that, again DNA polymerase can synthesize this part. So as you can see that DNA polymerase will synthesize this part, and the RNA primer will be removed and DNA will be there. And finally, the gap, the phosphodiester bond will be formed by one enzyme. This is called ligase. This is nothing but DNA ligase. Again, ligase in common term, ligation means to join two things together, like ligation. So similarly, the name of the enzyme is DNA ligase. DNA ligase. So DNA ligase will fill the gap. So as a result of that, now you have continuous DNA molecule, and this is not made up of RNA anymore. Like everything is DNA here. Now from one DNA molecule you are getting 2 DNA molecules. So that this is the basic mechanism of DNA replication. But there is some problem if you see that how this part of this DNA will be synthesized when we remove the RNA from this end, because again the 5 prime, 3 prime, the directional issue is there. So this is particularly a big problem for eukaryotic replication, because the eukaryotic DNA is linear. In case of circular DNA, it is not a problem. It will be synthesized. If you try to draw in pen and pencil, you will be seeing that it will get the 3 prime OH group and it will be synthesized. But in case of linear DNA, it is a real problem, that how this end will be synthesized. The linear DNA, in case of eukaryote, the end is called telomere. The end of this DNA or the chromosome is called telomere. There is some specific problems and some specific solutions are there in order to replicate the telomere, the end of the DNA. If you want to go in more details, you can follow any standard textbook. In this class, I am not going to discuss that. This is much more complicated stuff, but whatever I have discussed, this is the fundamental understanding, the basic understanding about the DNA replication which is happening inside every living organism, and that's all. Thank you very much.